Welcome to Module 2 of Sensation and Perception Online. This presentation will cover the basic anatomy of the human eye and how it transduces light energy into neural energy. It will also touch on the basic visual processing that occurs in the eye and ways that the eye adapts to changes in levels of ambient lighting. What we know as visible light is actually just a small, minuscule part of the spectrum of electromagnetic energy. As previously mentioned, our senses are limited to detecting only a small portion of the energies that are available. There are two ways of talking about light energy. The first is the photon, which is a measurement of the quantity or amount of light energy. The second is wavelength, which is a measurement of a characteristic of light energy. Looking at the figure, you can see that wavelength corresponds to the perceived color of visible light. We will discuss color vision in a later module. The human eye consists of multiple parts, and each has a distinct function. The first structure to come into contact with light is the cornea, which refracts or redirects light into the eye. The second structure is the iris, which is the colored muscular diaphragm that controls how much light enters the eye. It regulates the size of the pupil, the dark center of the eye that allows light to enter. Next, light passes through the crystalline lens, which is a pliable structure involved in visible focus. The ciliary muscles connect to the crystalline lens and control its shape. Finally, light strikes the back wall of the eye, the retina. The retina is a light-sensitive membrane that converts light into neural energy. The point on the retina that is directly behind the pupil is the fovea. It is the focal point of vision and the area where high-detail visual processing begins. This region is also referred to as the macula, as in the disease macular degeneration. The final structure that we will mention is the optic nerve, which carries visual information from the eye to the brain. The point where it exits the eye is called the optic disc. This area of the eye is not sensitive to light, so it is a blind spot. Let's zoom in on the retina and look at the neurons involved in visual processing. First, let's look at what is called the vertical pathway. It begins with the photoreceptors. These are the specialized neurons that transduce or convert light energy into neural energy. By neural energy, I mean the electrochemical process of action potentials and neural transmission. The photoreceptors synapse with bipolar cells, which play an important role that we will discuss shortly. The bipolar cells synapse with ganglion cells. These too play an important role in visual processing that will be reviewed shortly. For now, remember, the axons of ganglion cells are the ones that leave the eye via the optic disc, making up the nerve fibers of the optic nerve. The lateral pathway consists of horizontal and amacrine cells. These neurons are involved in a process called lateral inhibition. That is, the horizontal and amacrine cells modulate the activity of the bipolar and ganglion cells, improving our ability to see small differences in contrast. In other words, they help us see the details in this grayscale image of the retina. Starting at the beginning of the vertical pathway, the photoreceptors produce and contain a light-absorbing pigment that is broken down when struck by light. There are two types of photoreceptors. Rods are specialized for scotopic, that is, very low light, vision. They produce and contain a pigment that is very sensitive to light, such that a single photon can break it down. Rods are so sensitive that they cannot even function if the lighting is much more intense than moonlight. Rods are not found in the fovea, the center of vision. So, if you are ever trying to read a book under moonlight conditions, you'll have a difficult time. Cones are specialized for daylight vision. The pigment that they produce and contain is much less sensitive than that of rods, allowing them to function under an extremely wide range of lighting conditions. Cones are found throughout the retina and are densely packed into the fovea, so cones are not sensitive enough to function under moonlight, and they are at the center of vision. That is why you can't read in the moonlight. Color vision and high acuity, high detail vision is dependent on cones. Continuing along the vertical pathway, let's discuss the bipolar and ganglion cells. In the fovea, each cone synapses with two bipolar cells, an on and an off. The on bipolar cell responds when the cone detects an increase in light, whereas the off bipolar cell responds when the cone detects a decrease in light. Remember, our senses are change detectors. Each bipolar cell synapses with one ganglion cell. That is, there is a one-to-one relationship. As you can see in the figure, that means that there is an on ganglion cell and an off ganglion cell. This linking is incredibly important. The one-to-one relationship means that no information is lost in the network. Remember, neurons aren't sending information like status updates on Facebook. It isn't even Morse code. There is no content or added meaning to the transmission. We could think of a neuron's action potential as the equivalent of it saying, "Hi," to its neighbors. The only information is the number of times per second the neuron says, "Hi." An on bipolar cell says "hi" to its neighbor, the on ganglion cell, more often when its other neighbor, the cone, says "hi" more often. That is, when the cone detects more light. In contrast, the off bipolar cell says "hi" more often to its neighbor, the off ganglion cell, when its other neighbor, the cone, says "hi" less often. The more often the bipolar cell says "hi" to their respective ganglion cell, the more likely each ganglion cell is to say "hi" to its next neighbor, a neuron in the brain. So when the brain receives a "hi" from a particular on ganglion, it knows that more light was detected by that particular cone. So now that everyone is confused, let's move on. In the periphery, many cones connect to just two bipolar cells, and many bipolar cells connect to just one ganglion cell. What effect does this have on the neighborhood neural network? What it means is that when the brain receives a "hi" from the peripheral on ganglion, it doesn't know which of all those 9 cones detected more light. All it knows is that more light was detected by someone in that neighborhood. So, to bring it all together, a one-to-one relationship allows the brain to know exactly where changes in light intensity occurred. In other words, a one-to-one relationship is the basis of high detail or high acuity vision. A many-to-one relationship loses the detail, but is great for detecting light levels in general. That is, many-to-one relationships have higher sensitivity to light detection. Another aspect of retinal processing is the receptive fields of ganglion cells. A receptive field of a cell is the region on the retina on which stimuli influence the ganglion's firing rate. In the fovea, the receptive field is very small, whereas in the periphery, the receptive field is large. Remember, one-to-one in the fovea versus many-to-one in the periphery. Ganglion cell receptive fields are circular and divided into center and surround. These center-surround receptive fields underlie our ability to detect differences in contrast and involve the horizontal and amacrine cells, the lateral pathway. What you need to know is what they do. In the case of an on-center receptive field, when light falls on the center but not on the surround, then the ganglion cell fires. Notice in the figure that the yellow dot represents light falling on the center of the receptive field. To the right, where it says response, the vertical marks on the line represent neural responses. We can see only 2 responses before the yellow shaded area. Those two responses represent the ganglion cell's baseline firing rate. The yellow shaded area represents the time during which the light was on the center of the receptive field. There were a lot of responses during that time. Then, after the yellow shaded area, when the light is turned off, the ganglion cell returns to its baseline firing rate. Now see what happens when the light is presented into the surround. There are very few responses in the yellow shaded area when the light is on the surround, but then a burst of responses when the light is turned off. The decrease in light in the surround caused the neurons to fire. What about an off-center ganglion cell? Looking at an off-center ganglion cell, we see basically the opposite effect. No responding when the light falls in the off-center. But we see a burst of responding when the light is presented into the surround. My final note here is to remind you that this processing occurs in the eyeball. Visual information is processed and manipulated before it ever reaches the brain. This is a Hermann grid. Notice it is just a bunch of black squares in a grid. But do you notice anything else? You should see some gray dots at the intersections, but not the intersection that you're currently focused on. Why do we see gray dots when there clearly aren't any there? And why do they disappear when you try to look right at them? This is evidence of the receptive fields at work. Whenever you look at a particular intersection, the gray dot disappears because the fovea, the center of vision, has the smallest receptive fields. So, the white all falls in on-center receptive fields and is seen as white. But in the periphery, the receptive fields are larger, so some of the black squares falls in the on-centers, and some white falls into the surround off, causing us to see gray. Here is a case of our eyes reporting something to our brain that isn't even there. More importantly, even though we know the gray dots aren't there, we can't stop seeing them. We mentioned earlier that our eyes function across a wide range of light intensities. We've all had the experience of being in a well-lit environment and then suddenly being in a darkened environment. We also know that with time, we can start to see in the darkened environment, and with enough time, and if we're trying to sleep, we might wish it was a little darker. So we know that our eyes adapt to darkness. Here is how they do it. The first mechanism is one we are probably all familiar with: pupil dilation. When in a lit environment, the pupil constricts, reducing the amount of light that enters the eye. In a darkened environment, however, the pupil dilates, allowing more light to enter. But pupil dilation, as you can see, happens in less than a second, so that is only part of the story. Remember, rods and cones produce and contain photopigment. When the lights go off, there isn't enough photopigment available in the rods and cones to be sensitive to the low levels of light. It takes time to regenerate the photopigment. The figure shows the threshold, the level of light required to be detected, across time since the lights were turned off. The red line shows that cones regenerate their pigment and reach maximum sensitivity at about 7 minutes. It takes rods, the blue line, about 25 minutes to reach maximum sensitivity. The purple line represents the combined sensitivity of rods and cones. The delay in photopigment regeneration is what takes so long for us to adapt to darkness. Another important component is the center-surround receptive fields, which we've already discussed. Since these fields are sensitive to light level differences between adjacent areas, that is, sensitive to contrast, they assist us in seeing in a variety of lighting conditions, including dim light. In fact, they allow us to see in conditions that are dimmer than would be expected by measuring rod and cone activity alone. To sum things up, visible light is a minuscule portion of the electromagnetic spectrum. It is measured in photons and described by its wavelength. The eye redirects light into it, focuses light on the fovea, and controls how much light enters the eye. Photoreceptors in the retina, the rods and cones, transduce light into neural energy. They produce and contain photopigments that absorb and are broken down by light. The retinal neurons, the bipolar, ganglion, horizontal, and amacrine cells, form a network that begins processing visual information before it ever leaves the eye. The primary processing involves the center surround receptive fields, which enhance our contrast sensitivity. Finally, the visual system adapts to the ambient level of light through pupil dilation and constriction, through photopigment regeneration, and by using the receptive field contrast sensitivity.